Hello, and this lecture is going to be about cell division and cell death. So, mitosis is a process of cell division of somatic cells, and apoptosis is sometimes called cell suicide. It's also known as programmed cell death. It's a precise genetically programmed sequence of events which leads to ultimately to cell death. So mitosis and apoptosis mold our body, and they are very important for our growth, development, maintaining health, healing from disease and injury. And in our body, you always have an interplay between mitosis and apoptosis. So what you're seeing in this picture here is that in the embryo, apoptosis actually carves out fingers and toes from webbed structures. So in this particular picture, you can see that in this case, apoptosis actually failed to carve the digits, and the webbing still persists here in this picture, in this particular case. So before we get to mitosis, I wanted to speak a little bit more about apoptosis or apoptosis. So like I said, mitosis and apoptosis must work together to form a functional body. And when you have more mitosis and less apoptosis, that could lead to cancer. So in this particular diagram, you can see apoptosis at work. So an example of apoptosis is when our skin is peeling due to sun damage. So here's an overview of programmed cell death or apoptosis. So there are basically two pathways to apoptosis, and something called an intrinsic pathway, which happens from inside of the cell, and extrinsic pathway, which happens from the outside of the cell. So on the cellular membrane, you have something called a death receptor. It's very Star Wars, I think, like a Death Star. So here you have a death receptor, and a molecule called a ligand. A ligand is basically a protein or a small peptide or an amino acid. So in this case, we call it a death ligand, binds to the death receptor, and the death receptor gets activated because it's tyrosine, the intracellular domain that contains amino acid tyrosine gets phosphorylated. And once the receptor gets activated, it activates a series of enzymes called caspases. So caspases are enzymes that can destroy various proteins and cellular components, and then the cell disintegrates into something called blebs. So it actually falls into these fragments called blebs. This is also known as blebbing. And then the cell fragments get ingested by a type of immune cell called phagocyte. So here you can see cell components being ingested. So this is why we say that apoptosis is very different than another form of cell death called necrosis. So necrosis is when the cell ruptures, and actually it could cause inflammation to the surrounding tissues. Apoptosis is a type of cell death where it kind of goes away without affecting any of the other cells that surround it. So yeah, so it goes away quietly, and that's why some people say that some have called it like a cell suicide. So it goes away without damaging any of the surrounding tissues. So where does the name apoptosis come from? It comes from the Greek, which means leaves falling from a tree. Okay, it's a very poetic name. And so what are the main features of apoptosis? So we're not gonna go into a lot of detail, but just briefly. First of all, cells which are undergoing apoptosis become more rounded. So we have this rounding of cells. They undergo chromatin condensation. So their chromatin chromosomes are more condensed and more tight, and there's a reduction of nuclear volume. There's, like we said, plasma membrane blebs being formed, nuclear fragmentation, DNA becomes fragmented, and then the cell forms into these apoptotic bodies which get engulfed by phagocytes. And yeah, so in terms of biochemistry of the cell, on the surface of the cell you have a display of molecules called phosphatidylserines. So it's a type of a phospholipid which is present in the cell membrane. So it's called phosphatidylserines, and this way you can actually, there's a procedure that you can use to stain phosphatidylserines, so you can detect which cells in your, suppose you're looking at a cell dish with various cells and you wanted to count how many cells are undergoing apoptosis, then you could stain for phosphatidylserine. And you can also use a procedure called FACS analysis, which is a way of sorting cells. And so you could sort all the cells that and count the cells undergoing apoptosis. Okay, so another thing that happens in apoptotic cells, there's an activation of BCL2 family of proteins, specifically BAX, BAC, and BID, and those proteins are important in the intrinsic or intracellular apoptosis pathway. And usually in a cell undergoing apoptosis you have an increase in reactive oxygen species, so it's undergoing oxidative stress. You also have activation of enzymes called caspases which cleave other proteins and break up the cell into pieces essentially. And you also have a dissipation of mitochondrial membrane potential, so important to apoptosis. And yes, and also you have mitochondrial membrane permeabilization, meaning that the membrane becomes more permeable. Bax and back proteins actually form pores, or at least they form a single pore in the membrane, which allows for certain, specifically cytochrome C, which is another important molecule that we'll be talking about. So essentially mitochondrial membrane becomes permeable and lets out cytochrome C, which will activate caspases. And like I said, DNA fragmentation is another marker of apoptosis. So the take-home message from this slide, the slide just goes over the markers of apoptosis. So again, to summarize, phosphatidylserine exposure on the membrane, activation of BAX and BAC and BAD, increasing reactive oxygen species, activation of caspases, mitochondrial membrane becomes more permeable and releases factors like cytochrome C. There's a dissipation of mitochondrial membrane potential and fragmentation of DNA, and also the cell shape changes like you can see in this column. Okay, so this is a very complex figure of apoptosis. We don't have to go into this complexity, but essentially what it's showing you is, first let's look at the extrinsic pathway of apoptosis. Here you have the death receptor, and actually the name of these receptors, they are sometimes called a disc complex. And here you have a death ligand, which could be either TNF alpha or Fas binding to the death receptor. And so you don't have to memorize the names of the ligands, okay? And the receptor gets phosphorylated and activated inside of the cell. So its intracellular domain gets phosphorylated or activated, and that activates caspase 8, and caspase 8 is gonna activate another type of caspase called caspase 3, which is gonna actually lead to various events that cause apoptosis, okay, and finish up the program. So caspase 3 is called the effector caspase. Caspase 3 7 is another effector caspase because they actually do the job of cleaving the proteins and so on. Caspase 8 activates the. So another thing that's happening here in this extrinsic pathway, you can see that it also activates backs and back proteins, which assemble and form a channel in the mitochondrial membrane, which will also trigger the intrinsic pathway. So let's look at the intrinsic apoptotic pathway. Backs and back form a channel, and this leads to the release of cytochrome C molecule from the mitochondria. And then cytochrome C actually activates procaspase 9, which again is a type of caspase that later on activates caspase 3 and 7, which are the effector caspases. Okay, there are a bunch of other interesting proteins, like some of them have interesting names like SMAC and Diablo and BAD and BCL2, but we're not going to get into this whole pathway. But it's really a fascinating pathway to study, apoptosis, programmed cell death. And like I said, when you don't have enough apoptosis, it may lead to cancer. So there's a lot of cancer research is focused on how do you induce apoptosis in cancer cells. How do you kill them by apoptosis? Okay, and you can see that another thing that happens in the cell undergoing this process is its membrane potential changes because more sodium is flowing into the cell and more potassium is going out of the cell and more chloride is actually going out of the cell. Also, the intracellular amounts of calcium are increasing inside the cell. So you have all of this ionic gradients also changing as the cell is going through this process. So now we talked about apoptosis and how it has to be in balance with mitosis. So now we're going to talk about mitosis and cell division, but before that we should talk about something called the cell cycle. Cell cycle is a sequence of events associated with cell division. So the events that are happening before mitosis are called interphase, okay? So we have all of these phases that take place before mitosis are called an interphase. So interphase includes an S phase, a G1 phase, and a G2 phase. Okay? Some cells never go through this cell divisions, the cell cycle. Those are the cells that have differentiated and have specialized so much that they don't divide anymore. An example would be a neuron in our cerebellum or frontal cortex. So neurons that are very specialized and they're not dividing and they are in G0 phase. Okay? And so let's look at the cell cycle. The cell cycle is composed of G1 phase, S phase, G2 phase, and M phase, also known as mitosis. And as you can see here, mitosis itself is also divided into phases, like prophase, metaphase, anaphase, telophase, and cytokinesis. So okay, so what happens in S phase? In S phase, DNA gets replicated. All the chromosomes get copied. Okay, and G1 and G2 phases are when the cell expresses its genes, proteins are translated. And the one thing that I also want to talk about is that there are checkpoints when you go from one phase to another phase. So for example, when you're going from G1 phase to S phase, there's a checkpoint where there are certain proteins that make sure that the cell is prepared for DNA replication. And once you go from S phase to G2 phase, there are proteins that make sure that there is no DNA damage, that DNA have been replicated properly. So one of these proteins is called P53, which is a protein which is mutated in many cancers, and this protein detects DNA damage, and it usually signals, if the cell does have a lot of DNA damage, it signals for the cell to commit apoptosis. P53 activates the expression of various other transcription factors like P21, which will eventually activate other genes that will signal the cell to commit. Once the cell gets into the G2 phase, and then at this point mitotic spindle is starting, proteins that are going to be a part of the mitotic spindle are being produced, and mitotic spindle is being assembled. So there's a checkpoint here before going to mitosis, which is going to check if everything is present, if mitotic spindle is present, and if the cell is ready to go to mitosis. And so if there are any issues, again, the cell might not go into mitosis, and instead it might commit to programmed cell death or apoptosis. So again, there are checkpoint proteins like ATM kinase, which detect DNA damage, or P53 protein, which also detect DNA damage. Checkpoints also are present after mitosis. So you can see that there's another checkpoint right here, which ensures that chromosomes have been correctly distributed between daughter cells in mitosis. Okay, so now we're going to be talking about S phase. So S phase is when chromosomes are replicated. So here we have an unreplicated chromosome, and just before it gets replicated, chromatin gets condensed and DNA synthesis occurs, and this results in something called sister chromatids. So each chromosome ends up with a copy of itself. So you have like a duplicate of each chromosome. So each chromosome has its own sister chromatid, and the two sister chromatids are joined together by a structure called a centromere. So here we can see the centromere shown in blue. Okay, so for example, if you have chromosome 1, at this point you would have two copies of chromosome 1 joined at the centromere. Okay, so this is just another slide essentially saying the same thing. You have the chromosome, it gets duplicated, and you end up with two sister chromatids joined with a centromere. And this forms this X-shaped structure, but you shouldn't confuse this with an X chromosome. This is just two identical linear pieces of chromosome being joined together by a centromere. Okay. Yeah, so if you were to go to the previous slide, yes, so for the X chromosome you would also have the same thing, where an X chromosome would get copied, and you would have two sister chromatids corresponding to replicated to the replicated copy of the X chromosome and the other copy of the X chromosome. Okay. So what happens during interphase before mitosis? So DNA gets replicated, and there are proteins that form the mitotic spindle, and they get synthesized, as I said, during interphase. Microtubules are proteins that form structures called centrioles. So here we have centrioles being shown, okay? And the centrioles have microtubules, which are these long spindle-like fibers projecting from the centrioles. So just to go back again to the cell cycle and repeat what we said about checkpoints. So when the cell is in the S phase, S phase can last a different amount of time for different cells. For a liver cell, which is very slow at dividing, it could take up a few days to replicate its DNA, but for many cells, it's about like 8 to 24 hours. They could do this, be in this S phase. So basically, once the DNA was replicated, it goes through the DNA damage checkpoint, and if the DNA has been damaged or not replicated correctly, the p53 protein, ATM kinase, those types of proteins are going to inhibit the cell cycle until DNA can be repaired. Then it goes into the G2 phase, and that's where a lot of the proteins are being synthesized, which will be important for mitosis. And this is also an apoptosis checkpoints where again proteins like p53 might send the cell into apoptosis if there is DNA damage or if the cell is not ready to go through mitosis. But there's another protein called survivin which accumulates, and it makes sure that the cell can go through mitosis. So once it goes through mitosis, there's a checkpoint during mitosis which is asking, is spindle built? Did chromosomes attach to the spindle? Are chromosomes aligned down the equator of the cell? So again, if that doesn't work out, if there's some signaling going on saying that the spindle has not been built correctly, that can also send the cell back to apoptosis. Okay. So what happens during mitosis? During mitosis, the sister chromatids are separated into daughter cells, and so in the end, each daughter cell contains the identical genetic material. So if you had a diploid mother cell, so you end up with two daughter cells which have the same amount of genetic material. So I know in high school, students are often asked to memorize what happens in prophase, anaphase. This is just a funny slide. If you remember the old, now it's old, Twilight saga, and there's a scene where Edward the vampire and Bella are going over, they're looking at slides of onion cells going through mitosis, and they have an argument, is this prophase? He says prophase, she says anaphase. So I'm not going to ask you guys to memorize what happens exactly in each phase, and you should just be able to, you'll be presented with this information, so you should just be able to know just the basics of what's happening, because I know many of you know this material from high school already. Alright, so here we actually have this is a figure showing onion cells going through mitosis. So here you have late interphase. In late interphase, chromatin is condensing into chromosomes. Centrosomes have duplicated in preparation for mitosis. So centrosomes are, there's a lot of CE words like centromere holds sister chromatids together. Centrosomes are part of the centriole structure. So you can see the centrosomes are the structures from which mitotic spindles are being attached to the centrosomes, and they're all a part of the structure called centriole. So anyway, so here we have centriole centrosomes being formed. In early prophase, duplicated chromosomes are visible. Centrosomes begin moving apart, nuclear envelope is fragmenting, and nucleolus disappears. Nucleolus is the small organelle here. In late prophase, spindle is in the process of forming, and chromosomes are attaching to spindle fibers. And like I said, spindle fibers are made of microtubules, and here you can see microtubules are attaching to the, which connects the two sister chromatids together. And okay, so the next phase is called metaphase, and in metaphase duplicated chromosomes are at the equator of the cell. In anaphase, the daughter chromosomes or the sister chromatids start going to the opposite poles of the cell, and in telophase, the two daughter cells are forming, nuclear envelopes are reforming, nucleolus appears in each of the two daughter cells, chromosomes become more indistinct as they become less condensed, and then cytokinesis begins. So again, I wanted to go back to talking about just again the vocabulary. We have centromeres, which hold the two sister chromatids together, and centrioles, again microtubules form structures called centrioles, and they're kind of going out of the centriole structure. And centrosome is an oblong structure that organize microtubules into a spindle. So after telophase, the next phase is cytokinesis. So at this point, there is a cytoplasmic division taking place, which takes place after the nuclear division. So nuclear division is when chromosomes are going to each daughter cell, and you can see the nucleus is being reformed, nuclear envelope is being reformed. So now it's time to divide the cytoplasm, or distribute it between the two daughter cells. And then microfilament band contracts, separating the two cells. So there are hormones and growth factors that stimulate mitosis. First of all, what is a hormone? Hormone is made in the gland and is transported in the bloodstream to another part of the body. Think about for a moment, what hormones do you know? One example would be insulin is a hormone, or estrogen or testosterone, those are also hormones. So hormones exert a specific effect on the tissue. For example, testosterone stimulates spermatogenesis in men. So a growth factor acts locally. For example, epidermal growth factor, EGF, stimulates cell division in the skin beneath the scab. So if you have a skin injury, okay. So a hormone like estrogen can actually stimulate mitosis. So an example would be estrogen stimulating cell division in the uterine lining, preparing for the implantation of an embryo each month. Okay, so each month pituitary gland is gonna secrete LH, luteinizing hormone, and FSH, follicle stimulating hormone, which will bind to the receptors on the ovaries and signal for the ovaries to produce estrogen, and estrogen is going to be required to stimulate the uterine lining to lead to the mitotic division in the uterus. Okay, but here's another example. Estrogen might also stimulate follicles which will lead to the maturation of the oocyte, or basically lead to the maturation of the egg cells. And one egg cell is released every month while during ovulation. So here you have a hormone essentially being involved in mitosis. In the first example, when we talked about the uterine lining, that was an example where hormone is stimulating mitosis. But in the second example, where estrogen is stimulating the production of a mature oocyte, that would be an example of a hormone stimulating meiosis. Okay. Growth factors normally are involved in stimulating mitosis because they stimulate the division of somatic cells. So now we are going to be talking about another process called meiosis. And I'm gonna record that as a different lecture, okay?